it came about. It developed over several years, and I've been getting more and more comfortable with it, but I'm not sure I'm completely comfortable. So I'm really eager to hear anyone's comments about what they think and what we might have missed in this and whatnot. So, and the other thing I'd like to say is that a large number of people were involved with this. It's been a real group effort. So Jingwan Lee and Lydia Babcock Adams were students in my lab. Matt helps with the mass spectrometry, and we've really benefited from discussions with Lauren Mank and Matt Church at Montana on bacterial, iron-limited bacteria and genomics. So, okay, so next slide. There we go. Okay. So just as a brief introduction, for several decades now, it's been recognized it was a real paradigm shift when it happened, that over large parts of the ocean, iron is one of the key nutrients involved with phytoplankton and heterotrophic bacterial production, and that in many areas of the ocean, something like 30% now of the ocean's surface layer, iron is one of the key limiting nutrients. So those areas tend to be in the equatorial Pacific, the Southern Ocean, and in the western reaches of the North Pacific and North Atlantic Ocean. So as you go from north to south, down the Pacific, which I'm going to do, the nutrient that really is thought to limit primary production changes as you go. So when this was recognized, again, decades ago, the question came up is, how do microbes acquire iron from seawater? So iron's a bit different than other nutrients. Nitrate, ammonia, phosphate, they tend to be inorganic salts. They're small, they're readily taken up by bacteria, but iron is different. It doesn't really exist at very high concentrations at all as the free ferric ion in seawater. It's mostly complex to organic matter. So this became a really interesting question as to how microbes can get iron from seawater. And there's a number of different pathways that they can use. They can take up ferrous iron in the 2+ oxidation state directly and rather easily. It was thought that microbes evolved in a very anaerobic type ocean and with high concentrations of ferrous ion, and they have the ability to take up ferrous iron quite easily. Ferric iron in the +3 oxidation state is the dominant form of iron in the ocean today, because we live in a mostly or have a mostly oxic ocean. The solubility of ferric ion is really low, on the order of picomolar. And microbes do have specific transporters for ferric ion, but they're not that abundant, I don't think, and certainly not all microbes have them. And so microbes have evolved other ways to do that. One is just to reduce iron 3 to iron 2, which can be taken up, and this is done by cell surface reductases. Again, some microbes have them, some don't. And then another way is this production and uptake of siderophores, which is the one we've been studying. So siderophores are these very small metabolites, small organic molecules. They're synthesized by bacteria specifically to bind iron. They have a geometry that really binds iron very tightly. They're excreted into seawater. Once they're there, they can interact with iron that's bound to other organic compounds because of the strength of their that binding constant. They can take up that iron from the other organic compounds, and then presumably through diffusion make their way back to the cell. This some bacteria have specialized receptors and transporters for these iron siderophore complexes. They're taken into the periplasmic space and in some cases into the cell itself. The iron's removed and the siderophore can be recycled and excreted back into the environment to do another cycle. So it's a really interesting and common way to get iron in the environment. It's very common in terrestrial systems. It's common in some endosymbionts, cholera bacteria, for example, make siderophores. There's over 400 siderophores that have been identified and characterized to date. But of those 400, only a handful of the microbes that make them are marine, and only a small number of siderophores have been shown to come from marine organisms. So there was real questions about whether this was a viable iron acquisition strategy in the environment. And the difficulty is that it takes a lot of metabolic energy to make and to process siderophores. So if you think of, at least I do, most microbes in the environment are really living on the edge and are energy and nutrient limited. To do this whole pathway of making a siderophore, releasing it, taking it back up, taking the iron out of the siderophore and releasing it back into the environment just consumes a good deal of energy. And the other issue is that, once it's released in the environment, you may never see that siderophore again. It could just, they're made of very labile carbon and nitrogen. And the one on the left here is ferrioxamine B. It's a common class of siderophores. It's not found in marine systems that much, but other members of the ferroxamines are. And you can see they complex iron by binding through oxygen and nitrogen groups. This is a small peptide, and other marine siderophores are a combination of peptides and lipids. And so they're very energy rich, they're very labile, and if you're a bacteria and you're carbon limited and energy limited, these would make great substrates on which to grow. So when you release them into the environment, you may never see them again, and one fate could be just consumption for energy and nutrients. The other issue is that, in seawater, as I said, most of the iron is bound by organic ligands. There are tens of thousands of these in seawater. They vary in ligand strength, but to acquire iron, the siderophore has to compete with these ligands to get the iron. And finally, there are classes of microbes, and they've been isolated from marine waters, that they don't make siderophores, so they don't actually expend the energy to do it. But they can cheat and they can use siderophores. So you may produce a siderophore, go to all the trouble of baking it and release it to the environment, and somebody else may get the benefit of that. And in co-cultures where they have cultures, cheaters, and producers together, the cheaters always win. They always overtake the cultures. So there's a lot of siderophores, and for a long time it was unclear whether they were prevalent in the marine environment anyway. And a lot of that problem centered around methods that we could quickly and adequately sort of measure siderophores. And this is something I started working on probably 15 years ago. A small digression into it because it really impacts the experiments that we do later, interpretations. So the problem with siderophores is that their concentrations are low. We don't know what siderophores could be in the environment, and dissolved organic matter itself, there's tens or hundreds of thousands of different organic compounds in there. And so we don't know which ones could carry the iron. If there are siderophores, we don't know which ones are there. And basically, we didn't know how to find them. And so the way we found is into two major one was we just wanted to find the siderophores and quantify them. And our contribution to that, we took seawater, we concentrated the organic matter, which is a fairly routine thing. We did component spectroscopy. So this is just a tube. It's packed with beads. You apply your samples to one end of it, and you pump solvents and water through it. And the different organic compounds migrate down the tube at different speeds. So as they tube, they separate to some degree. There's tens or hundreds of thousands of organic compounds in seawater, so they don't fully separate, just separate different classes. In our case, we took the output of that and put it in PMS, inductively coupled plasma system. So what mass spec does, it burns everything up at high temperature. It measures the different elements that are the effluent that comes in the liquid chromatograph. And so what you end up with is a plot of time versus abundance. And so this time, so how long it takes these different compounds to come out of the chromatograph. In this case, versus iron 56. What you can see is, the is running, and then you get a sharp peak, this sharp increase through points in time. And so each of these peaks represents organic complex, and complexes are leaving the liquid chromatography system, allows us to the retention times, and it allows us to because the area under these peaks is proportional to the amount of iron in. And then the second thing we do is we get some chromatographic system, and you can do this in parallel or simply through sort of a conventional organic mass spectrometer, which just scans all the different masses from 150 to 1200. It does that every millisecond, it records the number of ions that hit the grid, and you end up with these mass spectra. Put the axis, and now mass is on the other axis. So we can go back to our that earlier slide, and I knew, for example, that something is at 40 minutes, and I can attention time, I can pull out the mass that are associated with that. So that gives me all the different organic compounds that came out of that moment. Again, there's thousands. And in the siderophores, what we did is we wrote computer algorithms that went through and queried each one of these masses, each one of these, and asked the question, is there another mass that's 1.995 Daltons heavier, in this case, and ask the difference between the iron 4 and iron 56 isotopes that are existent. And the thing was that any organic complex will complex both isotopes in about their crustal abundance. So it finds the ions that have pairs of ions mass separate. And then it asks the question, the ratio about 16 to 1, which is ratio of iron 6 to iron 54. And if it is, then it goes on to a target list of potential siderophores. And the output of that is simply, here's my trace of iron at the top, and then we find, in this case, 985, 983, that correspond iron 56 and iron 54 isotopes of the iron in that complex, and we scale them relative abundance. And that this peak has massive 95 and is most likely Merinobactin A. So that's how we find and quantify the siderophores and identify them. So with that in hand, we joined the GP15 Pacific Meridian Transect, a cruise run by the United States in Alaska and across the Alaskan shelf into the subarctic gyre, into the subtropical North Pacific gyre, the equatorial dwelling region, and into the top of the South Pacific subtropical gyre. Cruise that we're working now that starts into heat down to Antarctica, so eventually we'll have a whole Eastern Pacific cover. But this cruise track number of very chemical regime, coastal zones are productivity, high iron, subtropical gyres, and to the area, the high nutrient, high productivity, and low iron Pacific. And our thinking by bisecting these different systems that we would, in some cases, see siderophores produce when iron becomes limited, and other cases where there is high iron, we wouldn't see them produce. And so that was the thinking going into this. And in one side synopsis of it, we found siderophores in many, many samples. I think something like 80% of samples, they were picomolar and above. Position of the siderophore change. Sorry. Location, and we think it depends on the different possibilities of iron that characterize these systems. Concentrations in surface waters in the And I'll see the low air high subarctic gyre reached 70 or 80 picomole. And so it was really, it showed that, in fact, siderophores are common in marine systems. They're produced almost every by many different microbes and many siderophores, many of which we can't identify yet. But the real surprise in this was that most of the siderophores we found, the highest concentrations anyway, were in the pelagic, between 200 and 500 meters depth. And this is something that we didn't expect at all. We thought that, in the surface waters, especially in these HNLC regions, there's a real fight for iron. Iron concentrations are low. The potential for production is high. And that if you're going to use specialized systems to acquire iron, that's where we found it. We do relatively high concentrations in equatorial upwelling and subarctic gyre. In the Pacific and South Pacific waters, the system is very stratified. Iron's fairly high, nutrients are very low, and we didn't expect to find them. And in fact, most probably didn't. But temperatures drop, productivity falls because it's all being fed by this influx of labile organic matter from the surface for the most part, and iron concentrations begin to rise. And so that would be a reason siderophores. This is the iron siderophore concentration, something like 70 or 80 picomolar, actually represented only about half of the total siderophores in the system. Because if you remember back when I was describing how these things work, the bacteria releases a siderophore to the environment, and when it does that, it doesn't have iron in it, and it takes a while. In the experiments we've done, it usually takes 1 to 3 days for it to actually acquire iron. So there's this short period of time where the siderophore is in seawater, but it doesn't have iron. And because of the way we measure it, because we're measuring back here, we're measuring essentially, and if it doesn't have iron in it, we won't see it. And so what we've done for these is we take the sample, we analyze it, then we actually add iron to it. And what we usually see is a significant increase in the amount of siderophore. And it suggests that something like half the siderophores that are in seawater don't have iron in them yet because they haven't been in seawater long enough for that to happen. So this was a real problem. Like I said, we didn't expect to see this. And so we batted around different ideas. There's a number of them about why this might be. One of them may be that these siderophores are injected by sinking particles. So Phil and we've done some where if you think of these particles that you catch in sediment traps and even the flocculent material, the marine snow type material, these are really high carbon, high energy environments where bacteria are really going to town, metabolizing that organic matter. And if the heterotrophic bacteria have a high iron requirement and carbon respiration, the enzymes that catalyze that have a lot of iron in them, you could imagine a scenario where either the iron's lost from the initial substrate or just the consumers have a need more iron than they're getting in their diet. And so there's this real need for iron, and they secrete siderophores to compete for the iron that's in the sinking particle. We've done some experiments where we've incubated these particles. We've done a number of analyses, and we don't know the answer. I'm not going to have to go into it tonight. We think that was contributing to these high concentrations. The other one is just the microbes that live at these depths for the similar reasons, but as these particles sink, they dissolve and they inject labile carbon into the dissolved organic matter pool. That's the substrate for bacteria, and that their iron limited for similar reasons, in that concentrations are going up. But that's the concentration of iron. It really doesn't tell us how much of that iron is bioavailable. And it could be that the bioavailability of iron really changes with depth, and that the iron that's available anything between 200 and 400 meters just isn't what they need. So they make siderophores to acquire what they can. And so to look into that, we did some experiments, and these went on for several years. We did them at Station Aloha, which is located off the coast of Hawaii. It's near our geotraces station 17 and 18. Aloha is a very nitrogen limited system. Concentrations of nitrate in the euphotic zone are something like 1 to 10 nanomolar, and ammonia is at similar concentrations. And nitrate really doesn't come back into play until you get below the deep chlorophyll maximum, about 150, and then it starts to increase rapidly as organic matter is regenerated and are respired and the nutrients are regenerated. Iron at Aloha is supplied from above and below. There's a significant dust supply, and this is data from Jess Fitzsimmons. Did a whole series of cruises at Aloha. You can see high concentrations of iron in the very surface. They're high, but they're still fairly low. They're about half a nanomolar to 100 picomolar. It changes with season and with time. But as those dust particles are stripped from the water column, and as the iron that's extracted from them is stripped from the water column by export, concentrations fall and to right about the region of the DCM, there's a minimum of maybe 50 picomolar, and then they slowly increase as iron is released from sinking particles. So this is the environment that we did our experiment in, and this is our experiment. So our thought was, the natural system, we've got these bacteria making siderophores, picking up iron, and then using that iron. And what we wanted to do is to sort of tap onto that system by making our own siderophores. So we grew bacteria in the lab. We isolated and cleaned up these very specific siderophores, and we labeled them with iron 57. And so we knew which siderophores we were adding, we knew how much, and they were labeled with this iron isotope. And so we could follow these ones in the system. So then we add them to the system. And in principle, these should be taken up by the bacteria. The iron 57 should be removed, and the siderophore should be released back into the experimental bottle itself. And so our idea was that we would add these siderophores incubated and then collect the sample and analyze to see how much iron 57 was remaining. So that was the first sort of analysis we did. And because if these things were in fact cycling, we would expect that the iron 57 would be stripped out, but that the siderophore itself would be released back into seawater. And then we should be able to see these siderophores without their iron label because we know which ones we've added. So there's sort of two things going on here. One is the actual sort of uptake of the siderophore, and that's the loss of the iron 57. That could be for iron. It could also be that these were just being consumed as labile substrates by anybody, and therefore they would disappear from the system. So there's the loss of the siderophore, but then there's the production of these siderophores without the iron 57 in them. And the way we measure that is because we're only measuring iron is after we take the sample and analyze it, we add iron 57 back to the sample, and it'll recomplex with these siderophores. So if they're there, we should see an increase in iron 57 after we've done the addition. So here's the first part of that analysis where we added about 90 picomoles of iron 57 labeled siderophores. That's the gray line here. We incubate for 5 days, and then we analyze for the iron 57 siderophores, and that's the blue lines here with depth. So in the euphotic zone, we lost something like 30 picomole, about a third of the siderophores, and that continues right through to the deep chlorophyll max. But then when we get in the mesopelagic, right where we saw these high concentrations in the geotraces samples, effectively all the siderophores are used up. And then when we get below 400 meters, they come back. In this sample, they went away again, then they came back again. We don't know why. But in general, we've done this now about 6 or 7 times, and we see this loss of siderophore in the mesopelagic, and then it rebounds as you get deeper in the ocean. So that's the drawdown. So that's the total of respiration plus loss due to iron uptake. And this is how we can differentiate those two. So I'm going to start on the right here. So This is just a portion of that chromatogram where we're measuring iron, in this case, iron 57. So the siderophores we added are shown in gray. Each peak, each one of these peaks is a different siderophore that we've added. That's the filtered controls after 5 days of incubation. We take the experimental treatment, which is unfiltered, so we just let the bacteria go at it for 5 days, and we measure the iron 57 siderophores, and that's what you see in light blue. So the difference between those two are all the siderophores that were taken up for a combination of, labile substrate respiration plus iron. And then our third analysis is we add the iron 57 back, and siderophores which are there but don't have iron in them anymore should reappear, and that's exactly what we see. So the difference between the blue and the light blue and the dark blue are just the amount of siderophore that was in the sample, but the iron had been removed. So that's the siderophore that was used for iron. And that allows us to sort of partition the siderophore uptake at different depths between, not being used at all, used for iron acquisition, which is the difference between the blue line and the dark blue line here, and then the amount that was used for energy, which is the difference between the dark blue trace and the gray trace here. And what you find is in this mesopelagic zone, virtually all the siderophores are used, and about a third of them are used as just carbon substrates. We don't recover them at the end of the experiment, but about 2/3 of them we recover at the end, but they're missing the iron. So it shows that these bacteria took up the siderophores, removed the iron, and injected them back into the environment. And you can also see that in this bottom trace here. So this is aluminum 27. So aluminum, siderophores will also bind aluminum. It has a different ionic radius than iron. It doesn't bind nearly as tightly. It's about 2 orders of magnitude weaker binding. But there's a lot of aluminum in seawater. Concentrations are, I think, equal to or greater than iron itself. And so if these siderophores without iron are released into the environment and they bump into an atom of aluminum, they'll bind it. And so what you see here is, again, the gray traces at the end of 5 days of filtered incubation. So this is the control. But in our experimental treatment, we picked up a lot of aluminum in our siderophores. And for this to happen, these things must have been in seawater at some point without any down to any metal. And that could only have happened if the metal had been removed. So it's a real nice demonstration about that the bacteria in the mesopelagic are really cycling these things. The incubation is 5 days because we thought it was probably happening slowly. We do have other data. So we do this filtered control, but we also do a time 0 point, which I haven't shown here, but basically we filter the sample, we add the siderophores. We also have an unfiltered sample where we add the siderophores, but then we filter it right away and process it. And those samples are always about 10 to 15% lower in siderophore concentration than the unfiltered controls. And what it suggests that in the 3 hours it takes for us to process these samples, the bacteria have consumed something like 10%, 15% of the total siderophores. So what's happening is, there's a lot of bacteria. We don't know how many or exactly who yet, but they're excreting siderophores into the seawater. They can bind with either iron or with aluminum. That those siderophores are being rapidly taken up. The iron's been removed, and this cycle is going around and around. So it really shows that they are using siderophores to acquire iron. That experimentally, that really only happens under iron limiting conditions, and that's a whole nother talk. But it appears that iron is really could be really affecting the ability for heterotrophic bacteria in the mesopelagic to use the carbon substrates. When one way we thought about this was to looking at the carbon to iron, the nitrogen to iron ratio in bacteria that are grown under iron limiting systems and in bacteria that are recovered from, say, the surface waters of the subarctic where iron is the limiting nutrient. They tend to have a cell quota of nitrogen to iron of about 20 to 1. This is, I think, micromole to nanomole. And where we see these siderophores being produced is right where that the ratio of N to iron goes above that. And so we use that as a proxy. I honestly scientifically don't know what to make of that, because I'm not. I wouldn't think that bacteria living at these depths are need to take up nitrate from the environment. It may just be a proxy for how much carbon's being remineralized. But if you go around and you look through the section that we have about this N to iron ratio, right where, I can see if I can go back, right where that ratio hits the value of 20, which is the bacterial quota, it's right after that we see these siderophores really becoming abundant. So it suggests to us that bacteria in the mesopelagic are really being affected by iron concentrations. And having sort of stumbled upon that or ended up there, we've been looking through the literature about other possible indicators. So this is a paper by Mazzotto that appeared in 2020. He works in Max Thato's group here, actually next to me at Woods Hole, and they look at proteomics. And in this paper, they were looking at the expression of a tone B dependent iron transporter. So that's what this is here, this tone B dependent transporter that's used to acquire siderophores from the environment. And they too found a really high concentrations between 200 and 400 meters in this section that they did across the equatorial Pacific. And then if you look around, there's some other papers. There's a paper by Baltar. We've actually done some incubations of iron and looked at leucine incorporation at depth. And there are hints that it is stimulated by iron. So our thinking is simply that, in the surface waters near Aloha, we have, or even north of that, you've got very high productivity. There's high iron demand, but at Aloha, there's also high iron, so it's probably not iron limited. But once you get below the euphotic zone, there's this high carbon flux, there's high iron demand by the heterotrophic bacteria. Iron, at least bioavailable iron, is still low, so siderophores are produced to assist or facilitate the acquisition of that iron. But then eventually, when you get down to these depths of 400, 500 meters, the carbon flux is really attenuated. Iron concentrations are rebounding. Iron demand is low, and so this upper mesopelagic zone of siderophore concentration and utilization sort of goes away. So for me, the fun part was I never thought about nutrient limitations other than in the euphotic zone, but this suggests that might extend below the euphotic zone. We suspect that this occurs because of the slower regeneration of iron and perhaps due to a higher iron requirement bacteria. There's very little data on that, so we don't really know. We do know based on the 1,000 or so samples we've run so far that the siderophores are very common in the ocean. We also know that they turn over very fast, and in many cases, if you calculate the turnover time and the amount of iron going through this, it's certainly sufficient to support production. If you think of the ocean in terms of volume instead of surface area, much more than 30% of the ocean is probably impacted by iron bioavailability. And not a lot is known about how iron-limited bacteria, it affects respiration. We do know that certainly iron-limited bacteria or grow much more slowly, and they have a much lower carbon utilization efficiency. So more carbon is respired to CO2 and put into biomass when the conditions are iron limited. So it has the potential anyway to affect the depth at which carbon is regenerated and the amount of carbon is regenerated. And we just think below 500 meters that things slow down to a point where iron doesn't matter anymore. So that's it. And as I said, when I started, there was a lot of people that really helped out with all different aspects of this. And I'd just like to thank them.
Great. Thank you so much, Daniel. That was very interesting. We are running over time, but if anyone wants to stay on to ask questions to both Daniel or Tachiro. Sorry if I continue to mispronounce your name. Yeah, feel free to stay on and yeah, ask any questions. Anyone jump right out? No. All right, Daniel, I had a quick question for you. At the end of your paper, you mentioned how, we should be thinking about iron limitation in the mesopelagic with relation to iron fertilization that we're kind of thinking about. Could you just comment on that quickly?
I'm happy to comment on it. So we didn't expect this, and then we had it, and we saw it often enough that we thought it was real. And then there was like, why is it? And there are lots of different explanations for this. And then so we think it's real. And then I go, well, if it is, so what? Doesn't really matter. And there's very little data on that. And I think that, if you're, so I'm not involved with any iron fertilization work, but I guess my simple thought, and the reason why I put that in the paper, I go, well, if you fertilize the top of the ocean, you add iron, a couple of things are going to happen. Some of that iron is just going to precipitate, it's going to turn into iron oxide, it's going to be scavenged by sinking particles, and it's going to increase the flux of iron even at depth, not just in the euphotic zone. So in one sense, you could imagine it might fertilize the amount of bioavailable iron between this 200 and 500 meter layer. And if that does have, if bacteria really are stimulated by iron there, then they might just respire more of the carbon flux coming down to begin with. But also that might really affect the depth at which it's respired. I think respiring it at 200 meters is a lot different than respiring it at 500 meters if you're trying to sequester carbon from the system. So it's not something that I don't do that kind of stuff, and it's not something I really think about. But I thought all of you who work on these things might want to put that into your models or calculations, or see if that maybe it doesn't make a difference, but it could, or maybe it does. I just don't know.
Certainly interesting and food for thought. Thank you. Does anyone else have any questions for Daniel? No?
I have a comment. So, when we wrote this up, I was sort of 50-50 about whether it would make it to review or not, because iron limitation is a really fraught term. People have real different ideas about what limitation is and what it means. And in fact, it went through several review cycles, which I was surprised about, because I thought in nature you either make it or you don't, but that turns out not to be true. But a lot of the discussion, and it was a lot of fun with the reviewers, was, do we need new vocabulary for limitation versus stress versus deficiency, and what does iron limitation really mean? And so this is just we don't actually measure of change in production with iron concentration or iron availability. We just measure what appears to be a biomarker for iron limitation. And that, and people now are working on genomic markers about, the number of transporters or the number of this, and it really changes the whole discussion about limitation and what nutrient limitation means. And so it might be something to think about. That would be one way to critique the paper.
Yeah, very interesting. Yeah, I find semantics is kind of a problem in the other fields.
But people get really wrapped up in it, and it does have meaning. It does have meaning. It does have consequences.
Absolutely. Well, thank you. And Tiktosher, does anyone have any questions for Tiktosher? No? Okay, I do. I thought your presentation was really interesting as well. And I I may have missed a little bit because I had trouble with the recording, but I just wondered if you could comment a little bit on migrating organisms and how that kind of changes things for you.
Yeah. Sorry, please question again.
Sorry.
Please ask me again.
Oh yeah. I just wondered if you could comment on migrating organisms and how that you could see that changing the different phytoplankton communities, and if that even makes a difference. So they're coming up and they're going further down.
You mean about the surface phytoplankton community?
Yeah, and then going to depths.
Yeah, that is our next question. That is very important, but actually we don't have the surface samples.
Yeah.
Yeah, so now we are collecting the surface samples and from the other institute. So we will try it about the the relationship between the surface community and the sediment prep samples. Yeah, that is our next challenge.
It's certainly a challenge. Great. Okay. All right. Well, with that, if anyone else has any questions, feel free to raise your hand, but it looks like we're all set. And thanks for everyone for staying on for a bit longer. That was really interesting this month. So hope to see you all at the next JETZON Zoom call. Thank you so much.
Right. Thank you.
Thanks.
Sorry
Thank
for
you.
the problem.
Oh, no. Thank you so much.
Thank you.